Okay, wonderful. So I will start right away. I have only 20 minutes, and I have a lot to share with you, considering your education efforts. But first of all, I can completely agree that Valencia is a great city, and it's great to spend some time over there. So the topic of my talk today is called radiation variation and the perforosome. Because this is quite fascinating to me. And also the subtitle is here in bold, DNN Thermography in Exercise Physiology. So I will try to give you a glimpse into our research approach to work with thermography in the field of exercise physiology. Yes, and I'm based here in Mainz, Germany, where I'm working in the Institute of Occupational Medicine, but also in I'm associated with the Department of Sports Medicine in Mainz for research and teaching obligations. Yes, the core and the key question that is the base for our research is, can infrared thermography support diagnostics in exercise physiology? And can it enable contact-free prognostics concerning individual physiological responses during exercise? And for all experiments, this is the key question, actually. So the first part of my talk is now on radiation variation and the perforosome. And for the start, I brought some videos with me from our recent studies. And you see on the left side on the screen, you see a person running in constant load, and you can observe how the skin temperature changes during the running bout here. And if you compare it to the beginning, now during the running, it actually decreases. The tendon region is kind of glowing a bit more, but the rest is decreasing. And then also in the end of the exercise, during the load, there's also the appearance of a thermal distribution pattern over the whole posterior legs. And this thermal distribution pattern, of course, is the thing that also made me so interested in this technique. And on the other side, you see a video, also constant load. It's a shuttle run test here. And you see directly an advantage of working with thermography, because you can screen actually the whole body and also several parts at the same time of the body. And you see again, there's the thermal distribution pattern over the whole body here. So that was one of our first observations when we started to work with thermography, that this pattern appears over the whole body during running, during cycling, and also during rowing or other endurance exercises. And the fascinating thing is that this pattern, this thermal distribution pattern, is quietly looking like blood vessels under the skin or beneath the skin. So there seems to be the possibility to look beneath the skin with the thermography. And now I have another example for you where we applied thermography during exercise testing. This is a graded exercise test, a RAM test for just 8 minutes. So it's a RAM protocol without breaks. And in the end, after maximum exhaustion, we did a verification board. And here you see a homogeneous temperature pattern over the whole legs. And then a slow start, but in the end, you know, you need to go all out then. You see the temperature decreases more, fast running. You see some superficial veins here as well. So there's directly some interruption here in our region of interest from other patterns. And then after the bout, you actually see the appearance of the thermal distribution pattern again. So the difference to the constant load is that we had no pattern during the running exercise, only in the recovery phase. And then when we started again with the verification bout, so it's in submaximal load, but it's close to the maximum, you see that the pattern immediately disappears again. And then it appears again in the recovery period. To make this more clear for your eyes, it's better to look at these images in resting images. Here you see the preconditions again, homogeneous pattern, then the decreased temperature. These are 3 different individuals in the recovery phase, there is the temperature pattern, and it's like a fingerprint. It's different for these three individuals. Then the verification bout, again, almost maximum load. You see the pattern disappeared again, and after the recovery, it appeared again and looks quite the same as in the first recovery period. So it's so interesting. It's an individual pattern. It's quite related to the individual anatomy of their own blood vessel structure. The question was, what do we see actually here? What is the structure? How is it called? And I looked up some studies here, and I found nice documentations from the research group from G.I. Taylor, who they actually doing research in plastic surgery, but they show these structures nicely in their publications. And that's called the perforasome. So you see there is one source artery, and there are different branches, and you can see these branches look quite the same as what we see on the surface of the skin. So actually it should be possible to locate source arteries based on the perforasome that we see on the surface. To have a more detailed look and directly see what's so difficult on measuring this pattern is that the source arteries branch up until to the surface of the skin, but they penetrate either through the muscle or in between muscles. So there could be a layer between or not. And that's also a reason why these vessels appear differently on the body surface when we measure it with thermal imaging. Here, just another documentation on part B of their documentation from the studies. The femoral artery, for example, from the cross-sectional area of the thigh, you see the source artery, and then through the tissue branching up until the surface of the skin. So this is more the anatomical description, but the question is, why do we see actually what we see during exercise? And as you know, when you perform exercise, you have the mechanical load, and you also have metabolic strain there. And here I just share with you some data that we recently collected during constant load or interval load measures throughout nearly an hour. Under thermoneutral conditions, you see that the core temperature rises up to 2 degrees here in our experiment, measured with a pill, an ingestible pill, or also ANT+ sensors. So the body needs to perform some heat loss mechanisms. And you know, there are many ways that the body get rid of heat. So on one hand, you have the heat gain factors. These heat gain factors in laboratory conditions under thermoneutral conditions are mainly due to contracting muscle and mechanical work, and also to metabolic energy expenditure. But then you also see, on the other hand, the heat loss mechanisms. All of us are aware of evaporation, but there are different or other factors like convection via muscle blood flow, or conduction or convection via airflow, and also convection via skin blood flow. And this convection via skin blood flow is, from our point of view, quite accelerated if the body vasodilate the blood vessels and the cutaneous arterial perforator vessels that we see with a thermal camera. And that's the reason why we see these vessels during endurance exercise in constant load. The question is now, why do we don't see these vessels during all-out exercise and first after the recovery period? So taken together, kind of a summary here, we have these resting conditions, nice homogeneous and no pattern. And then after quite intensive exercise, we have this quite reduced temperature. And the reduction in temperature is related to sympathetic noradrenergic nerve activity. And this activity actually increases when we start to exercise. And this is mainly to supply the active organs with oxygen during the exercise. And actually the body fine-tunes this nerve activity during the load actually associated with the external load to supply the muscles, for example, with oxygen. The second mechanism, when we see the vasodilation of the cutaneous arterial perforator vessels, is the reflex neurogenic vasodilation. So the vessels vasodilate to activate the mechanisms for heat loss. And then afterwards, in the recovery phase, when we stop the load, especially when we go all out, there's no more need for the sympathetic nerve activity here. We can, or the body can just reperfuse all the vessels and try to come to homeostasis as under resting conditions soon. The most interesting part for us is here that there seems to be a competitive situation for the body. On the one hand, supplying the active organs with enough oxygen and kind of constrict the vessels, and on the other hand, vasodilate the vessels to get rid of the increasing heat in the internal tissue. So this is quite fascinating. But the question then was for us also, can the local skin temperature variation of a certain body area be associated with a specific physiological trait during cycling? And that's why we did a quite nice experiment with our friends in Valencia. And this was also interdisciplinary with computer scientists and partners from physics. And we applied an experiment where we did a pyramidal exercise load protocol, and we measured several physiological factors like sweat rate, core temperature, heart rate, RPE, and also, of course, watts. And we applied 3 cameras at the same time to capture 5 different regions of interest in the face, in the forearms, and the calves. And to make it more quick, you can read all these results in the studies published, but you see that there is a different response in these different regions of interest. So there's quite a difference, and maybe we can relate it to different traits. So we found out that, for example, the forehead is the most associated parameter with the with sweat rate, but as you can see, the conditional R squared is 0.64. That's nice, but even higher is the relation of the calf's temperature and the heart rate, or the RPE. So you see the nice pyramidal load on the one hand, the heart rate, and on the other hand, you see the calf temperature on the right in dark red, quite in line with load. So there's kind of an inverse association here. So the calves seem to be a quite good marker or region of interest when it's about to discuss the prognostic value of thermography, for example, heart rate or oxygen consumption. So coming now to the part 2 of the talk, we talked about now about radiation variation, and also you know what I mean with the perforasome and the anatomical structure. But we also wanted to measure this temperature pattern, and there was actually no possibility. So we designed an approach that is called DNN, and DNN stands for Deep Neural Networks. So it's a quite computer science activity here in exercise physiology. Why do we need to do such thing? From our point of view, there are many possibilities, as you know, to measure skin temperature in several regions of the body. And it depends on how you select the region of interest, what is then the result in the end. And there are so many different, you can have one point, you can have the whole tie, you can have the anatomical shape, but none of these options capture actually, for example, the thermal distribution pattern. And also our colleagues from Italy, Perpetuini and Merla's research group, they also showed that 60% do manual region of interest selection, which also causes a marbled research landscape that makes it hard to conduct meta-analysis in our field. So, and the next aspect why we need this software or DNN approach is that you can have the same region of interest and you can have the same mean temperature, which in this region of interest. However, from a physiological point of view, it seems to be quite a different situation that the individual has before and after an exercise bout. And that's why we thought it must be really relevant to measure these different temperature or the thermal distribution patterns. So could we develop it? We had, again, an interdisciplinary approach with my colleague, Daniel Andres Lopez. He is a computer scientist, and he, together with him, we designed this deep neural network to capture the body parts and to capture the different thermal patterns, for example, cutaneous arterial vessels, superficial veins, and also have an automatic time series analysis of these data. So from 25,000 images that we captured during such one exercise bout, can we automatically analyze it? And here you can see an example on how it looks to measure the bodies automatically and then receive data from it from the deep neural network. Here you see some the normal image, and then you see on the right hand the patterns that are detected from the software, and then you also see the data below this video. And coming quite to the end of this bout, you see that there is a increasing temperature pattern, and this can be captured with the software now. So the question is, can we now synchronize the DNN thermography with other measurements of cardiopulmonary and thermoregulatory measures? And again, the same project, we set up our lab here, and you have a glimpse into our lab work now on the treadmill. And here you can see on the computer the first synchronized system capturing the distance of the legs, the angles of the legs, thermography, and also our spiroergometry measures, core temperature, and heart rate. Of course, we need a big computer for this, a thermal camera, time of flight camera, and also kind of a speed measurement system. That is how it looks like in the Department of Sports Medicine, by the way. And based on this synchronized system, we are now able to really have a look on synchronized time series data. And you can see here, for example, I just need to do my, you can see here on the top, there is a constant load bout, then there is a constant and interval load bout, and then there's intervals and constant load bout. And we compared these. And there you also see 4 different graphs. For example, in green, the perforating vessels. And you see, especially in the recovery period, they are much higher than, for example, the mean temperature or just the non-vessel area temperature. On the right hand, you can also see the core temperature, and you can also see, for example, oxygen consumption and heart rate during these 3 bouts. So now, actually, we have the foundation to extensively investigate the application of thermography in the field of exercise physiology, synchronized to our classical parameters like cardiopulmonary parameters or thermoregulatory parameters. So to sum it up or wrap it up a bit, we talked about radiation, for example, the thermal distribution pattern, and this is relevant to investigate thermography in exercise physiology, meaning not looking especially at absolute temperatures, but more to relative temperatures before, during, and after an exercise bout. Also, DNN thermography enables automatic, objective, and continuous measurement of skin temperature and different thermal distribution patterns like cutaneous arterial perforator vessels in resting and moving legs. And also now DNN thermography, I already mentioned this, is the foundation to extensively investigate these different distribution patterns and kind of investigating the relation and the application to be a non-invasive diagnostic marker for patho or physiological traits. So now also the measurement system allows a synchronized time series analysis for the first time. That's already the end of my talk. So thank you very much for your attention, here are the reference if you would like to have a look into the studies and all the other sources that I just used. And there is a video that we did for Medica. It's a big exhibition here in Germany, in Düsseldorf. There you can also find out more aspects. And then there are some great books also from education point of view. I would definitely recommend to have a look in these. Yeah, so contact me if you have any more questions, or there may be is some time for questions now. So thank you, first of all, very much for your attention.